and featuring Dr. Julio Palmaz, one of the 2019 recipients. The NAE and Ohio University recognized Dr. Palmaz and 4 others early this year for innovations in coronary angioplasty, enabling minimal invasive treatment of heart disease. The Russ Prize is currently the top engineering prize in the world, which is comparable to the Nobel Prize. You may not know this, but there is no Nobel Prize for engineering. Russ College alumnus Fritz Russ, who was an inventor who is an innovator in biomedical engineering himself, and his wife Doris established the Russ Prize with a gift to Ohio University. Their goal was to recognize achievement that improve human condition. Awarded every other year since 2001, the prize has honored accomplishments including kidney dialysis, the automated DNA sequencer, and the technology behind LASIK surgery and the cochlear implants. We are very, very proud to celebrate the engineers, scientists, and physicians who we call creators for good behind these technologies. They have saved and improved millions of lives. Now to introduce our guest. Dr. Palmaz is Chief Scientific Advisor of Vactronix Scientific and an honorary Ashbel Smith Professor at the University of Texas Health Science Center in San Antonio. He developed the first balloon-expandable vascular stent to receive U.S. Food and Drug Administration approval. In fact, the Palmaz stent is on display at the Smithsonian National Museum of American History in Washington, D.C. It has been used in nearly 100 million patients worldwide, making the Palmaz-Schatz stent one of the top 10 medical device patents of the last 50 years. Gosh. Dr. Palmaz joined the University of Texas Health Sciences Department of Radiology in 1983 as Chief of Angiography and Special Procedures until 1999, then served as Chief of Cardiovascular and Interventional Radiology Research until 2005. He was inducted into the National Inventors Hall of Fame in 2006, selected for the Gold Medal of the Society of Interventional in 2007, and inducted into the National Academy of Inventors in 2013. He is a distinguished scientist and fellow of the American Heart Association, a fellow of the American Institute of Medical and Biological Engineering, and a fellow of the Society of Interventional Radiology. Dr. Palmaz holds 60 patents and has authored more than 35 books or book chapters and more than 100 peer-reviewed publications in scientific journals. Without further ado, please join me in welcoming him to Ohio University.
Make me tired to hear all that in terms of, it's been quite a few years. At any rate, receiving the Russ Award was probably the tallest feather I have. It is far more meaningful because I always wanted to be an engineer myself. But somehow life took me to medicine, but I came back, I was able to come back through molecular biology and physical chemistry, so definitely I thought that this was incredibly flattering that I ended up getting an engineering award. At any rate, one thing I can really give you, particularly for the young people, if I can get somebody, one person in this audience stimulated to go into what I'm trying to develop next, is I've done my job. But definitely, since I've been there from the very beginning, the idea occurred to me in 1978, and it were so many years, and how my perception of all this field changed over the years and my expectations changed. So now I can, almost 35 years, 40 years later, I can give you a good past perspective. And perspective and perhaps cast some projections towards the future. I know what should happen soon, but the far future is what is exciting. So this is a curve of my own perception about the rate of achievement of progress in this area of let me see if I can turn up this phone. Yeah, the rate of achievement in cardiovascular medicine. And I would say that the 80s were an exciting time from many points of view, not just medical devices, but we saw the development of a new specialty, interventional cardiology, and things were going so fast that we had great expectations. Nonetheless, many of those expectations got tempered later on because, to me, when the drug-eluting stent came about, and I thought that as a poor solution to the problems of these new devices, adding immunosuppressive drugs to solve the problems of essentially local rejection. I thought it was actually abdicating to the challenge of biomaterials. The transcatheter aortic valve implantation is a great idea. It's rather obvious because it's a combination of pre-existing technologies. I will go about that around. I think it is a wonder of medical ingenuity, and I will make my point, a failure of industry. And then the bioresorbable stent, to me, was the low point because essentially they failed, got their FDA approval, and predicted it just failed in practice. So, so when we talk about money and how much money is going into cardiovascular device technology, the 80s were a wondrous time. Companies and corporations, big money was coming in droves and ready to battle with each other for preeminence in the field. And, and definitely the big corporations were ready to get in head first. And they all had long-term objectives. They all understood that there was a challenge, that the technology was disruptive and it was poised to replace traditional technologies such as surgery. And they saw a big future into it. They knew that they were undertaking a big financial risk, and actually they were essentially betting their shop on that. But the important thing from our point of view, it was broad-based. I mean, it was a class of new technologies, and that casted an enormous inspiration in all of us that were behind it. Things changed in 2005, and that's the year that I would put the inflection point where all of a sudden all the financial burden went to small startups and venture capital money. And these people are there to make money, and that's the only objective they have there. And they do it at the least risk affordable to them. The objectives are very short term, typically 1 in 3 years. You cannot do anything in 1 in 3 years because it's less than the approval time for any device. So what they can do, only incremental improvements, and that's essentially no breakthrough technology. Financial risk is, measured in dollars, typically is about from hundreds of thousands to a few $1 million, and definitely the focus is narrow, not broad-based. Cardiovascular technology, when we now look at the perspective of the past 30 years, is an adaptive technology, meaning that we didn't develop anything, really. We're using pre-existing technology, pre-existing materials, and adapting them to what we are doing today. Surgical wire and hypodermic tubing are the building block materials of most stents used today. And this is a 50-year-old technology, metallurgy-based technology that has not changed, but in few details. And then the rest is just equipment that was there before, like wire braiding equipment, 3D printing, lasers, CNC tooling. Essentially, we're using stuff that is already there. When companies come and say, we are breaking into new frontiers. That's not true. I mean, they're used adapting what is already there. In fact, the things are not even done in the US these days. Most of them are done abroad with equipment that is actually discarded from the US. And another issue with the development of this technology is that where the idea originated is very simple. I mean, I was author of the first one. You know, a scaffold of arterial lesion. I mean, of course, we put a balloon there, inflated it, deflated, the artery collapsed, the patient has a heart attack. So obviously, we had to put something in there to keep the artery open, a scaffold. So the timber tunnel principle was essentially the inspiration. And then we had problems with excessive healing response, restenosis. 1 in 4 will have that. So what do we use? Essentially, a drug to suppress proliferation, inflammation, and that was the drug-eluting stent. Today is where we are. To me, it was sweeping dirt under the rug. We never quite understood what was going on. In other words, never understood the primary response of the body tissues to the biomaterials that we were using. But we went ahead and used a suppressive technique, covered the stents with plastic at the beginning, and then put a drug into the plastic. And then the other one was the bioresorbable stent. To me, that was a sheer disaster. It was based on an assumption. I mean, Dr. Colombo, my dear colleague and tremendously inspired clinician, said it, doing the job and stepping away. And he wrote an editorial with that name, but it wasn't true. I mean, but at that time, the community was not even ready to hear arguments that if you put a scaffold system is to replace a function that is largely gone, the spine is gone, you need to put something that will forever replace the function that is lost. Nonetheless, some sort of magical thinking was that as the stent will vanish away, new tissue will appear, and the structure will reorganize in a functional way, and it didn't happen. So it's a term that I found that it's actually fits very well. It's called heuristic approach. Heuristic means that when you have a series of events that need to be proven before you embark in a scientific process, in other words, a rational thought, when there is a gap of knowledge somewhere, you apply an assumption, and that is a heuristic thought, all right? Used a lot in cardiovascular devices. One of them was a gold-coated stent. Somebody decided that they were going to coat and at that time it was 316L stainless steel and cobalt chromium with gold, because gold is a noble metal, isn't it? It's a good stuff. And it works well in the mouth, so it should work well in the vascular system. So it got approved, actually in a relatively fast track, 2001 by the FDA. And then it was quietly taken away from the market because we had a delayed thrombosis rate of about 25%. It was a disastrous device. The other one was the coronary stent covered with permanent polymers. At the beginning, again, I said, why we care about the biomaterial? As long as it's strong, let's use it. And if there is any reaction from it, might as well cover it with a paint. We put a permanent polymer on it, and then we make that polymer elute drugs. And that became the gold standard. All right, the problem is that there is no such a thing as a perfect cover. The thing will delaminate. They will have the trap fluid corrosion issues. And then, of course, the ions eluting from there will cause delayed tissue reactions and so forth. Nobody uses permanent polymers anymore. And again, approved 2003, they were the gold standard for many years. And unfortunately, many, many patients still walk around with one of those. The other one is the bioresorbable stents that I said, and I already mentioned the issues regarding that, but it was another heuristic approach because the medical community was absolutely enamored with the idea. They thought they were going to solve all the problems, and they even told the patients, why are you going to have a metal stent when you can have something that will go away? And so it was quite embarrassing, actually. So the other thing is, many of the new products, and the cardiac valve is today one of the latest, greatest accomplishments in interventional cardiology, the replacement of aortic and mitral valves with prosthetic transcatheter valves. But it's actually a combination of old technology, a bovine valve, or horse pericardium, combined with a regular balloon-expandable stent, Dacron, Teflon sutures, and so forth. So essentially, these things are like a piece of jewel. They have thousands of steps, and serial manufacturing is the thing. You have to see the places where they make these things, passing from one operator to the next, and stitches and cuts and fittings. And when it's done, the device is worth about $25,000, and it has hours and hours of manual labor on it. Or hardly what you would think of a medical device. So I think we need to change a little bit the way we approach these things. I think it's time for a change. It should have happened a long time ago. So we need to stop thinking of the cell as the central element in relationship to our devices. We need to think more on that we should actually emulate the extracellular matrix. The function of the extracellular matrix should be the inspirational point. And essentially, we should extend our level of observation. Our target should be a submicroscopic level, at receptor level, not at cell level. In other words, what happened with the cell is not enough for us. We need to understand what happened with cell receptors. And essentially, from the practical point of view, we should try to create habitats which are structural and functional for a determined cell population we are trying to address. So what are the properties of the extracellular matrix that it'd be interesting to emulate? I mean, with so many times in science we have done that, trying to emulate something like protein synthesis and the way genes work in ribosomes and things like that. I think the structural and physiological support for cell populations, essentially we're building real estate for cells. We need to mine the intercellular communications, which are quite active. We need to also find the right mechanical modulation so that the habitat is proper for the cells that we are trying to address. For example, bone, rigid environment, and of course, as fluid as it could be in like plasma. And we need to mind the segregation of incompatible cell types. Obviously, these cells are subject to immunological attack, and that should be something we should all be minding too. So I'm thinking like tissue engineering at this point. There's a whole bunch of papers that appear in the 90s that set the direction. This should have been stimulated hundreds of people going in this direction. Instead of staying in the simplistic way we are today, we should have looked into smart surfaces. And this came from the group of, I think, at MIT. And now, all of a sudden, there's a flurry of activity. 2 or 3 papers have come out in the past 3 years. And I think they came up with things that were astonishing to me. For example, if you put an endothelial cell in a field of adhesive proteins like fibronectin or vinculin in a non-adhesive territory, such as hydrated silica, for example, all of a sudden the cell stretches, trying to reach these adhesive dots. In doing so, the cell all of a sudden changes its metabolism in a dramatic way, which could be beneficial. For example, the apoptotic rate, the rate at which the cell dies, it has a sweet point here. You see, when you get to a pattern of 5 in 10, meaning every other double the space as the size of the dot, and on a certain size pattern, the apoptotic rate drops dramatically to almost 0. In other words, what are we doing? Making now the cells survive longer. Is that something good? I mean, definitely should be, isn't it? So there's something to look at very seriously. The other thing is that we use plasma cells of the same line, and we modify the extension of the adhesive territory. We can make that cell become different things. For example, in this particular case, an adipocyte in a small territory, you stretch the territory, it becomes an osteoblast. So that means all of a sudden we can modulate the nature of the cell, starting from a primordial cell. That is serious business. And finally, the other one also came in 1994. So this is already pretty old information. The function of hepatocytes changed tremendously insofar production of albumin, depending on how much you force the hepatocyte to stretch on the adhesive territory. When you went to this size, all of a sudden, again, there's a sweet spot in the distribution of adhesive sites. The production of albumin grew almost 5 times. That is, again, together with the survival and together with the differentiation capabilities, all of this is almost like wondrous, let alone drug elution, isn't it? So what do we have instead? I mean, the first years after we started with stenting, and we were in the plumbing business, essentially putting stents in arteries, we started looking at them with different ways. At the beginning, we used regular microscopy, it's worthless. But then we got the convolutional light microscopy that you can see depth very well on surfaces. And all of a sudden, we started seeing things like this. This was not like an army of cells marching on top of a stent. It was more like a stampede, where the cells were essentially going around areas. And this is a crystal, all right, a large crystal. As you can see, that the cells are crowding in this area here, they're avoiding this area here. And why they're avoiding it, all of a sudden, we became very interested in this. Is that something implicit to the nature of the material structure? Then we got interested in how the endothelial cells move on surfaces. And it's an incredible poetry of expression of adhesion sites. These are clusters of integrin receptors. And they express on the ventral surface as the cell rolls like a water bag on a surface. And they pick them up on this dorsal aspect. They express them on the leading edge. And then you can see here this lameripodia, essentially an extension of the cytoplasm, expresses all these adhesion receptors. And then they pick them up on the rear as they move forward. So essentially, it's a very highly orchestrated. You almost think that it's an animal with its own brain, the way it works. They have a strategy to move around. And so we started doing research in our lab and trying to get better definition methods to see these cells. And these are endothelial cells on chromium oxycarbide, which is pretty much any metal surface. And we can see this cell here expressing all these lameripodia on the front end and pick them up on the rear. So I'm walking happily, and this one here couldn't find anywhere to attach. And through this lameripodia all the way equal to about 3 times its own length, because it couldn't find a single attachment place. So that means that the distribution of attachment sites were incredibly irregular, despite the surface looking shiny and flat. So then we started looking at the chemistry of these metal stents, and we found an enormous desegregation of the alloys on the surface. For example, with enormous chemical heterogeneity. Here, for example, this is 316L stainless steel. You see, when we do an iron scan, there's a hole here, and the hole now corresponds to chromium in the chromium scan. Mean there's a separation of the 2 elements. Obviously, they're both going to have different types of chemistries and different oxide concentrations, and the cells are going to be sensitive to that. So the other thing was the topographical variability. I mean, the commercial stents are typically shown nice and shiny, looking like a real medical device. In reality, they look more like a moonscape, holes and craters and valleys, and particularly on the inner surface, which is not controlled, which is the useful surface for us. So in other words, a lot of problems, not just depending on chemistry, but just the structural stability. So as I said, a lot of the crude old machinery is being shipped to places outside of the US. A common place for stents to be made today is Ireland, Kuala Lumpur, and Malaysia, so forth. And of course, they do them there for a problem of cost, so they decrease cost. They come to the States already packaged, ready to sell. And so, we could do a better job, but the problem is that these things have become commoditized. I mean, there's a price limit that is worth for the companies to invest into it because they are not making much money. Competition has made these devices cheap. But the problem is that we are not progressing. So I think right now, if I'm going to propose a change in the status quo, and here is what I hope the new generation is going to have a dent, I would use a scientific approach and justify the goal without heuristic gaps. Second, try to restructure regulations. We are still based, as you remember, the original scientific thought was mechanistic. Now in medicine, we went to so-called proof of benefit, meaning that it doesn't matter how it works. The important thing, benefits the patient or not, but that's expensive. All right, today to have a new coronary stent in the market is about $1 billion if you want to go through the problem trying it, because you will have to actually prove equivalency within a range of under 1%. All the stents are doing well. All right, so I think we need a complete change in the manufacturing systems if we are going to go the real way that we should go, which is minding sub-micron structures. And essentially, just not think more scientifically, just not the interface with tissue and blood. This is a paper I published, I think it was my third paper on this subject, and it was 1993, it's 26-year-old. And at that time, this was my conception of what was happening from the tissue, blood cell and tissue interaction with a surface, which at that time, I knew it was oxide, at least for the materials that we were using at the time, was 316L stainless steel. And I was very interested in what happened in the first 7 to 10 minutes, because I knew that defined essentially what was going to happen with the stent later on. So whatever happened in this very early age, I knew it was critical, and I knew fibrinogen had a lot to do with it. So all of a sudden, fibrinogen became our workhorse. I mean, we were totally interested in it. We couldn't convince anybody else. But to me, fibrinogen is important for a very simple reason. It's incredibly abundant. There's 3 grams of fibrinogen per liter of serum, of plasma, and compared to milligram quantities of any other more interesting adhesive protein like vitronectin and fibronectin. Those are milligram quantities. This is 3 orders of magnitude more. Second, fibrinogen is an incredibly versatile and a smart molecule. It has quite a few ligand sequences which are recognized by all the critical cells we are interested in. All right, for example, this is the gamma domain, this is the alpha C domain. So you can see that right here in the main connector, we have attachment receptors for platelets, monocytes, fibroblasts, platelets, endothelial cells, and so forth. So all these are not expressed at the same time. It depends on the conformation of the molecule, how the molecule bends, how it changes shape, and it can. This is like it has its arms and moves around and is incredibly versatile. But obviously, it addresses the possibility of attachment of all the critical components on the blood reaction to a stent as we place it in. So for example, polar and nonpolar interactions are the most important ones. So meaning nonpolar is hydrophobic. That's the way glues work, because it's all short-range interactions, typically hydrogen. All right, so the alpha C, the gamma domains are largely hydrophobic, and they quickly deconvolve and make a nonpolar interaction with the surface if you have one. All right, and if it does that, conceivably expresses the attachment receptor from the alpha C domains, the RGD receptor and the ligand, and that would actually promote the attachment in the endothelial cells. In other words, the endothelial cell could walk on a surface covered with fibrinogen that has in itself attached to a hydrophobic surface. A polar interaction actually it leaves the molecule it gives more freedom to interact with other cells. So if the alpha domain interacts electrically with a polar negative charged surface, then all of a sudden the molecule can deconvolve and be recognized by a whole bunch of different actors, which may or may not be beneficial. So then you can start elaborating from there. Okay, so if we have that, then obviously, conceivably, we could have different conformations. You have a polar negative or polar positive or non-polar surface, like I mentioned. But all these things give you now a new element of judgment, gives you a molecule to test them, and now you're thinking within the size of the fibrinogen molecule. Fibrinogen molecule weighs about 125,000 Dalton, and its size is within about 500 Armstrongs, which is 50 nanometers. And so remember, it's 1,000 nanometers to a millimeter, so it's a large molecule. But that gives us a size scale to what kind of surfaces we should engineer. Now we're talking about engineering of surfaces at nanoscopic level. Very different from what we have today in industry. So what are the different strategies? It's not just that. I mean, obviously, charge would be if we can have here like a alternating positive and negative polar charge, and here comes a molecule that has both. We can actually capture both of them. If we, based on hydropathy, in other words, the affinity for hydrophobic or hydrophilic interactions could be based on that, or even in conformation. In conformation, actually, we could borrow the ideas from this thing, the MIPs, which are molecular imprinting polymers. In reality, it doesn't have to be polymers. This is a misnomer, because some of these MIPs are actually on oxides. And since we're dealing with metals, if we could use that approach. MIPs actually is a great way by offering essentially based on this steric conformation, we could capture molecules which are in parts per billion on a surface. In fact, this is used for water filtration to capture, undesirable chemistries out of water. So, and interestingly enough, it's not being used in medicine that I know of. It's also used in sensors, because smelling sensors, taste sensors, and so forth for industry. So, okay, what is the predictions then? I think a high definition manufacturing is important. We need more validation, and I will go on into a minute. And we need new stock materials and practices. Definitely, we need to abandon the stupid wire and tubing. I think it's a thing of the past. It's time to move out of the steam locomotive age. Now, what is fun is to look in the distant future, and that was the second part of my talk. And so to get you in the frame of mind, this now is highly theoretical and a lot of fun to think about it. It may never happen, but just think about the possibility that we could create structures on a chemical basis, ideally injecting these materials in the circulation. All right, so if we could create a nanoblock, which I call structural nanomolecular blocks, or SNBs, or in the order of a large molecule, like fibrinogen is, 100 to 800 nanometers. So relatively, it's still submicroscopic, but large molecules. Then implant the site of interest with reactive groups that will recognize those if you give them the correct chemical reactants. Then assembly with chemical reactants that could change by different means, enzymatic or pH or whatever reason, so that the complementary sites will meet and form larger structures now inside of, all right? And I think at this point now we are talking about millimeters or larger, and this probably will be monitored by imaging. And then a final assembling using external field energy, such as gamma or microwaves or whatever else. But so this is just to kind of a teaser just to think of that possibility. So let me make a quick review of where nanotechnology is being used today. Definitely it is already medicine, but we use it as drug carriers, nanomolecular tags for diagnostic imaging quite a bit. We use it definitely in biosensors, and I'm not going to get into that. For electronic neural interface, for motor cortex and retina, cochlea. It's largely experimental, but we're already there, but nowhere to be seen in cardiovascular devices. So why? Well, because all the materials that we use for as nanoparticles, they are not robust. These are materials that do not have structural properties. They are mostly like cadmium, selenium, zinc, sulfur, phosphorus, bismuth, arsenate. I mean, really, none of them are robust materials like transitional metals are, that you can build things that are very strong, all right? So this thing didn't start too long ago. It's called controlling the geometry of nanomolecular constructions. And this started about mid-2000s, and 2005, 2007, the first papers appear. The first were gold nanoparticles with chemical reactants placed at geometrical defined sites. Therefore, now you can have geometrical lattices. That was the first step, and it was actually quite an advancement. Then they tried other things, but this is based on barium sulfate, hardly a structural material, so forget this. This was a step forward. It's called viral capsids. Viral capsids, actually, which is glycoproteins, hard glycoproteins, they are very geometrically regular, so you could actually put reactant sites within it, and then you see this do things like this, change direction 90 degrees and move on and create structures. So this was a possibility. Of course, this whole thing from the virus of tobacco, mosaic virus, and things like that. But this is where the money is. The Rice University Fullerene, that is a magnificent new tool. I'm surprised that they haven't gotten into this themselves. But now we're talking about something that is strong, that's robust, chemically stable. You can make the derivatizations very easily. You can put any chemical group you want on them, and then you can make them join each other exactly at the point where you want them. And so here you can see, how you can open up any of the valences of carbon and replace it by a reactant group, and then have side chains doing anything you want. So it's non-toxic, widely available, cheap, and here carbon-60, the most common one, is exactly like a soccer ball, and in a structure, and it's only 1.1 nanometers, very, very small. But you can actually make structures. I mean, the first nanoblock could be made out of fullerenes put together outside of a body, and then if you have a smart nanoblock, then there would be something that could do what we dreamt about. Something like that. I mean, this is one thing I found somewhere, but it looks like a nanoblock made out of fullerenes with chemical reactants make them get together could look something like it. So I say that the critical steps for something like this, and hopefully a bright young guy with his mind into nanomolecular constructions could build it. You have to design and engineer one that will have and that's where the engineer mind comes, the ability to create other structures, have the design flexibility such as to create anything that would, in large size, mimic organic structures. Then obviously they will have to have a function, a site that is functionalized, but it would become active upon command by chemical reactants. And then you will have to have first, the activation of the site where you want to implant them,
and essentially build structures based on reacting coupling sites. So essentially, you'll be making a Lego piece, and it might end up looking like this. I mean, there is already some interest in all this area, and who knows, some people may be secretly working on it, not publishing it yet. So how then, I mean, as you put these things, everything seems possible, but there is one unknown factor, is time. You don't know how long, because we're dealing with very small sizes, isn't it? So, you know, the ability to create microscopic structures to something that small will be dependent on essentially the specificity of the reactions. If this happen at random and they don't happen very often, then it will take a long time to happen. So I think the idea would, for one thing, you had to work ex vivo until you get to about this orders of magnitude in size, all right? Then it gets introduced in a patient, and at this point you need to use technologies like massive parallel assembly so that these things happen in parallel, other than, you know, waiting for them to happen at random. And possibly at that point you will be in the microscopic realm. And you know, when you're about, you know, 6 to 10 orders of magnitude in size, then at that point it may be visible. So you start be able to monitor those things by imaging. So I think that, as I said, this would be a drug. This essentially would be introduced in a patient using the proper reactants, and then the monitoring, and then, as I said, external field energy may complete the job. The only other things that happens is that anytime you deal with a complex structure, and this is again an engineering question, you will have an enormous task on design. All right, so I couldn't find any better example than showing, okay, this little parrot, I mean, you can barely tell that it's a parrot, and it probably has about 10 Legos. And the instructions to build it is about maybe 10 bytes. It's very simple, isn't it? If you want to get something that looks a little bit more like a bird, you probably already like 6, 7 orders of magnitude more complex. Okay, this is already a brochure. All right, to be able to put it together, it will take a kid quite a bit of time. But if you want to get something that really look like a real thing, you know, with the degree of definition of the real thing, now you're talking about 4,000 terabytes. It's an enormous amount of computer. You couldn't do it definitely with information that comes in a brochure. You would require a computer assistance to be able to put it together. Well, imagine that same thing will happen when you're dealing with biological structures. So just to tell you how the measuring things has become complicated with time. When we came out with the first stent, the Palmaz stent, we used this thing. It was called an optical comparator. It was nothing but a big loop and light, and you were able to put cursors and measure. The amount of data that we got out of this was very small. Okay, but that was enough for the FDA purpose at the time. By the 2000s, we came into deconvoluted light. That is very important because it measures depth. And now we got into the sub-micron scale. So we can measure, you know, hundreds of nanometers. And these days, now we are using for some of the devices we make for optical surgery, we use X-rays. This is a nanoscope, the tomoscope. And we are measuring nanometers now, real nanometers. So for example, this is a polygon image of the device, and you can see now we measure, this is almost 100% validation of the device. But if you want to have something really precise, and creating a structure as complex as an organoid will be in that realm, it will require something like this. So 1990, when we started with the stent, the amount of validation that was required was 3%. In other words, we measured 3% in any direction in the device, and the FDA would check it as good, assuming that everything else was okay. All right, and the amount of the data we produced was about, it could fill in a piece of paper. All right, maybe 1 kilobyte, I think it was, of information. By 2000, we went to 5%. That was an exponential increase. Now, any stent, when it's used for AQL, it will be about 1,000 kilobytes, or 100 kilobytes or 1 megabyte. Right now, 2018, we are again under 5-micron resolution, but we had 10% sampling on the device, and we are at 10 megabytes. You imagine the amount of data that we need to collect to be able to qualify these things. That every 10th device needs to be measured, and this amount of data had to be collected and kept pretty much forever. And I would assume by 2030, when we get in high-definition manufacturing, look how complicated this becomes. The amount of AQL would be about more than a quarter of the whole dimensional spectrum of that device. Will be having tolerance limits in the order of 200 nanometers, which is what is going to be required if we want to do something really, really effective, and will be in the terabyte range. At that point, there is no cloud that can actually absorb all that. We are already having an outside data farm to be able to put the stuff that we are doing today. So a couple considerations. I'm amused for some of these things that people come up with, but this technological singularity defined as artificial superintelligence that will elicit boundless evolutionary growth. They think that by creating AI and incredible computing power that we are going to, the consequence will be evolutionary growth. I think it's wrong. It's the other way around. The evolutionary growth will elicit incredible amount of computing capability or need of it. So it's going to get very complicated. So to finish, I think intervention of the future may be at the control of the computer, and the whole thing will be chemical-based. The patient will be probably sitting in some sort of fluid bath, just like we see in the movies. And, these blocks will be introduced intravenously, probably, and the computer will tell us and be able to let us monitor what we are doing, all right? But I think it will not be like that. I think it's going to be more complex than that. I think making a kidney or a thyroid organ will be more complex than making a super heavy 787. It's going to require a whole control center with a whole bunch of different specialists, or maybe one in charge, but molecular biologists, engineers, physical chemists and chemists, supercomputer scientists definitely at the center of the whole thing, and a nanomolecular architect. All these people will essentially, this would be a very complex task. So what we see today for a NASA center may be the place where we are going to be able to do these things of the future. So again, about fantasy foretelling the future. This is already happening. So this movie, The Edge of Tomorrow by Tom Cruise, where Tom Cruise is clad with this battlefield apparatus that allows him to run at high speed, carrying enormous weights and weaponry. We are already aiming in that direction. We are actually working on brain interfaces to be able to pick up electrical signals, to be able to eventually do servo mechanisms to control limbs. And that is going to happen. One of these days you'll see it on the papers, and this is happening now. But this, getting to this point, I think this movie is actually pretty interesting. If you can go through all the fantasy issue, the details that the movie maker put in here are actually somewhat credible to me, at least, But Scarlett Johansson here is a semi-synthetic cyborg, and whatever biomechatronic is. But essentially, she is largely replaced by semi-synthetic tissue. So essentially, this is what we're aiming, in the future. Right now, we're having what we have today, hip prostheses and dental implants will look like, things of the past. We were going to have smart things being replaced, and semi-synthetic muscle and so forth, I think is a reachable goal, but just definitely reachable, not where we are today. We need to do a complete change mentality. So, so this to finish. So I love this quote from Theodore Sturgeon, a science fiction writer that died in the mid-'80s. And what he says here it clicks so much with my mind. Inner space is so much more interesting because outer space is so empty. In reality, I never found any reason to all the money we spend in outer space exploration when there is so much of the inner space that is not explored yet. Thank you very much.
Thank you so much for the exciting talk, Dr.
Yeah.
Palmaz. And it's very good to learn that our engineers really contributed to this great invention. So now this lecture is open for questions. So any questions?
You have some microphones. If you raise your hand if you have a question.
Any questions?
Thank you, Dr. Palmaz. Oh, there we go. Thank you, Dr. Palmaz. Very interesting talk, and I'm really happy to see so many of our biomedical engineers and our chemical engineers in the audience today. So, of all the questions that I would like to ask, I think the one that I'm most intrigued to hear your opinion on is that medical suite/lab of the future that you imagine, where you have a specialty-trained MD with an engineer, a molecular biologist. Do you imagine that to occur in our academic healthcare environments now, or are you imagining that this would be something we would see in a commercial space?
We spent lunchtime talking to the students about that. I was hoping that they were going to come up with the answer. Yeah, you're right. It is a monumental task, to be able to do these dreams of, cybernetic cyborgs. In other words, semi-synthetic tissues, implants and replacements. I think it's going to require enormous effort. I don't know if it's going to be government or it's going to be industry. Maybe industry, even if they can see it, and think a little bit more than they're thinking today, which is very childish mentality they're using. So I hope somebody will see the light. They did, back in the 80s. Hopefully it will happen again. But it's a large task. Now, what is the role of, like you said, of the individual in all that? I tell you, I'm ready to see that the first nanoblock will come from a weird place in the world, like a place that we don't even know by name. Because there is, first, because it doesn't require big equipment. All this is actually tabletop equipment that is required to be able to build these things. It's all chemical based. And second, it just requires open mind. Just like the kids are do better with the internet and computing than advanced scientists, simply because they have that boundless imagination, and they're thinking out of a box, in other words. So I'm hoping that one of these days we're going to see something like this. The kid said, "Oh, look, I've come up with this nanoblock that I can make it do this if I inject this, or do that if I inject the other chemical." But that would be the beginning, and then somebody will see. I mean, from there it's easy to trace the way, and so that's what I'm hoping. Some of you will come up with something like that.
That was a great talk. Thank you. This is a question coming from someone that knows nothing about stents. Other than when the stent goes in, the stents that are used today, it goes into a vessel. What happens to the tissue on the outside of the stent, not on the luminal side? Is it necrosed or?
Yeah, By the time somebody qualifies for a stent, the wall of the artery is largely gone. All the elastic structures are gone. All these delicate structures the artery has, they're replaced by fibromuscular tissue and calcification. So it's essentially garbage.
So there's no way to think about repairing that on the outside, so that
Yeah,
it
but
would
on the
reconstitute
outside,
the normal
yeah,
vessel.
you have some buildup of fibrous tissue. The adventitia definitely reacts to the presence of the foreign body, and you have neovessel formation and some fibrous formation. But the end is always like a metal covered by fibromuscular tissue and endothelial lining. That's a goal today, what we have.
Thank you for coming out. Maybe I missed this in the lecture, and if I did, I apologize. But do you imagine the next step in stents, before we maybe delve into nanotechnology, to be something that is largely just ignored by the intravascular space? So just a stent and a scaffold that is not collecting and growing a thrombus, or something that is just coated by endothelial surface or endothelial cells and kind of facilitates the growth of a new tissue that is then somewhat similar to normal?
Yeah, no, excellent question, yeah. I mean, you use the key words, largely ignored or highly reactive. That's the way with the stuff that we have at hand today, Dacron, Teflon, polyurethanes, all the transitional metal alloys and so forth, that we have that. All right, some of the materials like Teflon, all the fluoropolymers are the same, are ignored by tissues for a while. Then the tissues, the macrophages start figuring it out. Some protein, rogue protein, anchors onto the surface, then it comes a macrophage, then establish a colony, and then they start forming stuff, and all of a sudden they think thrombosis. And that's the typical fate of the Teflon implants, that happen with the PTFE grafts and the femoral popliteal bypasses. They do well for 6 months, and all of a sudden, boom, overnight they thrombose. Metals are a little bit more predictable because they are reactive. Therefore, they form this layer of cells, and then eventually, hopefully, the endothelial cells will colonize. But at the beginning is what we call the fight for the surface. The cells that are quick and that go fast, like the platelets, in seconds they found their attachment receptor and they plant in there, they send signal to other things like macrophages to come in. Macrophages are slower, they roll, and beta lymphocytes and B lymphocytes and neutrophils are slower too, but they're all circulating. And eventually, it's all who gets to the attachment receptor first. That's why it's so important to engineer a surface so that we eliminate that fight. Right now, it's random, and it's whatever gets there. We don't even know. I have my suspicions that even there's variability from stent to stent of the same manufacturer. So that creates this chance that you may do well or not. If you put a drug-eluting stent, then you do better because you're killing everything that plants there, isn't it? So, platelets can attach, but macrophages and they just cannot become migratory and, end up producing, proliferative tissue. So essentially, you create a poisonous field. But that is not a principle for implant technology. Implant technology should be friendly to the tissues, and you should control and modulate the response in such a way that is ordered and logical. So you'll get your fibrinogen, it will be and other adhesive proteins, then you will have the cells that you want colonizing there and staying there, so that they end up with a huge thickness, a big layer, which is the battlefield of all these species that, trying to predominate. So essentially to create predictability.
So
Yes.
from the engineering point of view, basically at the Russ College, we have a top corrosion lab. And this group kind of studies like the fluid, how they kind of like impact the corrosion problem of the metal pipes. And basically we know that if we have a tint or we have a dent on the metal pipe, and that area is more intent to cause corrosion of the metal material. So now we talk about blood vessel. So within the blood vessel, and you have a stent implanted. So that part will kind of like disturb the blood flow. So basically cause a turbulence. So for that area, is that more intent to have the blood component deposition?
Yeah, I mean, believe it or not, there is no such it's not a science, and definitely the plumbers, you know, the interventional cardiologists, use very simple principles, you know. They just try to overexpand a little bit, anticipating that it'll be, 5-10% luminal loss by tissue proliferation, and hope that that tissue will have endothelial cells. Therefore, it will be functional endothelium and it will be non-thrombogenic or antithrombogenic. But in reality, there's not much control. What you do is you try to achieve an open lumen. That's about it. And measuring, the flow speed in the vessel is the only thing you do. In other words, they open the faucet, see how much water comes out of it, and then you think the patient is all right. And then, of course, with dual antiplatelet therapy and things like that, you kind of manipulate the outcome. But it's not very scientific at all.
Yeah, so if we can think outside the box, or outside the stent in this case, the stent has to have a terminus. So the stent goes in, and then there's going to be normal endothelial cells at the end of the stent, right?
Right.
By definition.
Right.
So has anyone tried to engineer, perhaps right at the terminus, very minute amounts of VEG, vascular endothelial growth factor, or IGF-1, to allow those normal endothelial cells to grow into the lumen of the stent?
Tell me again. I'm not sure I follow your idea.
Okay, vascular endothelial growth factor.
Yeah.
VEGF, okay?
Yeah.
And it promotes endothelial cell growth. And that's what we want in this case, right?
Yeah.
So at the terminus, where you have normal endothelial cells at the junction of the stent, to have at the ends, at the termini, very small amounts of this VEGF release to promote the growth into the stent. Would that be possible?
Well, they've done it. They use that. They found out that it tends to be thrombogenic because many of those things are non-selective and there are other things coming in. The closest thing to that idea is putting progenitor endothelial cells in the stent, and they are using that. Not the endothelial cells themselves, but they put attachment receptors for progenitor endothelial cells, on the assumption that they are plentiful in circulation. Therefore, when blood starts going by, that endothelial cells that are circulating will come and attach. Well, it turns out that a lot of those supposed to be progenitor endothelial cells ended up being macrophages, so they think really that technology did not move forward. They're still there, and they're still, trying. It's very difficult to compete against the drug-eluting stent because the revascularization rates of drug-eluting stents are very good, very, very good. About 5%, 6 months. So they give enormous protection, really, I have to admit. So in that respect, particularly for high-risk people like diabetics with small vessels. But it's very difficult to come up. Orbis is the name of the company that is doing the progenitor endothelial cells. But good luck to that. I mean, at least it doesn't have drugs, which is, to me, an advancement. And the idea is not bad.
Thank you for coming and giving your talk. I think that's very good for our young students to hear this. Mine doesn't relate so much to technology, but I help run the biomedical engineering program and work with undergraduate students and graduate students all the time. And it's quite clear to me from listening to your talk and your success that you've certainly found something you're passionate about. You're very passionate about your science. I imagine you're in a field that you really love. What role did mentors or your parents or your friends play in helping you find something that you were passionate about?
Leave me alone. That was the best thing he could do for me. No, I mean, no, I kind of, it's one of those things that, you know, it was totally serendipity, you know. I mean, I love the project. I had nothing to lose, really, Abby, because I was a resident in the radiology program, and somebody was paying my salary, and so I just went home and played with these things. I had a lot of fun doing it. But at some point, you know, the more I got into it, the more enthusiastic I became, and I always said I'm not very smart, but I'm persistent, and I think that served me well. Yeah, I just stay with it. Yeah.
Thank you so much, Dr. Palmaz.
All right, thank you.